A microbiota may consist of bacteria, archaea, fungi, viruses, and protists. All of genetic material in a specific ecological community are called microbiome. Recent studies in the microbiome have developed rapidly, and researchers have found the microbiota could have a profound impact on plant and human health. The following figure can serve as a proof of incremental trend of peer-reviewed articles in the last 20 years or so in the field of microbiome studies that includes both human and plant sciences. Most of the research work is conducted in human than plants. Several studies have shown that microbes in human body have a great impact on immunology, digestion, absorption, and other physiological activities. It is believed that there might be two-way communication between brain and the intestine. This gives us an opportunity to try to apply similar concepts in plants. Currently, the focus in studies of plant microbial communities is shifted from initial descriptive phase towards mechanistic understanding of the microbial assembly and functioning of microbiota, the functional role of individual components, and the possible application in the integrated plant management plans. It is of great significance to understand the composition and function of the microbial community for different diseases or physiological states, which will greatly benefit the disease diagnosis and their treatment. There are two basic questions to answer for the microbial studies: who's there and what they are doing. This is not easy as it seems to be, because the microbiome is complex, highly diverse, and dependent. It is estimated that there are more than 1,000 kinds of bacteria living inside the human gut, and this number may not be different in case of plants, thanks to the next generation sequencing technology that enabled us to see the microbes inside these systems. With the help of next generation sequencing technology, or NGS, we could retrieve all the genetic information in the sample. NGS technology provides us with opportunities to understand the composition, function, and dynamic evolution of microbial communities. However, it is often limited by metagenomic data analysis methods because the metagenomic data is too large and complex to be studied through visual means such as correlation analysis. This hampers our deeper understanding of host-pathogen interactions. Oh, but wait for a moment. Why microbial studies are important for us? One of the main goals of the microbial study is to explore the relative abundance or diversity of microbes. Then there is a need for finding the association between microbes and diseases. And number three is to analyze the different states of the disease to lay the foundation for further application in the follow-up treatment or forensic identifications. Now we know the importance of studying microbes. Alright, so the next question arises: when we are studying microbes, how about the data that is being collected? How do we analyze it? So for analysis, researchers start to utilize machine learning methods to craft the relationships between microbes and their hosts. It is believed that machine learning could learn and discover patterns from the data. However, the performance for each algorithm is usually dependent on feature engineering, which is the process of using domain knowledge to extract features from the raw data. When performed manually, the process of feature engineering for machine learning can be prone to error. Here is a schematic diagram of a general process that how machine learning methods utilize microbial data for the analysis. So as you can see in this diagram, that apart from raw data, there can be external data that has been fed to the machine, and then it is learning itself and evaluates the model. Now that we know how the data is fed to the machine, it is very important to distinguish between deep learning, machine learning, and artificial intelligence. So deep learning, also called neural networks, is a subset of machine learning, and machine learning is a subset of artificial intelligence. Machine learning function starts with manual extraction of significant features from pictures. Here you can see this photo that usually a data scientist is required for the extraction of features. This is a cumbersome process since, as you can see in the photo, there is a car. So a data scientist has to directly feed some features from car photo to the machine to give it instructions. In contrast to this, when automated feature engineering is used in case of deep learning, none of these obstacles exist. That makes the process faster, and also it reduces the time. As we discussed earlier, that deep learning is better than machine learning in the case of microbial data analysis. Deep learning could discover highly complex relevant features to improve prediction. Commonly, three applications of deep learning in microbial data analysis are as follows: metagenomic classification, metagenomic gene prediction, and lastly, microbiome wide association studies or genome wide association studies. We will discuss them one by one, but first we should define what is metagenomics. Metagenomics is the study of genetic information in uncultured organisms obtained directly from the environment. Before diving deep into the applications of deep learning in microbial data analysis, I would like to give you a brief overview that deep learning actually mimics human brain. At its core, the concept of deep learning is relatively simple and mirrors a similar process by which humans use information, experiences, and trials and errors in learning. People accumulate historical experiences and generalize these experiences to speculate on novel problems, where underlying assumptions or processes may not be known to predict specific outcomes. The training and predicting process in deep learning can correspond to the human generalization and speculation processes. Just as experience is important in learning, historical datasets play a decisive role in deep learning. Within the data analytics field, deep learning is an approach that is used to design complex models that implement themselves for prediction. In environmental or ecological studies, these analytical models allow researchers to output reliable, repeatable results and discover unknown relationships through learning from these historical datasets. Alright, let's get back to the examples of deep learning application. The first one is metagenomic classification. To characterize the diversity of the microbial community is one of the main objectives for metagenomic research. Here I will be presenting you important applications, which are three, of deep learning in microbial data analysis. The first one is artificial neural network. This method is used where the input is supplied in the form of numbers. So for example, self-organizing maps is a model which is used in artificial neural networks. The second one is called deep learning deep neural networks. So in this there is a convolutional neural network or CNN. This model is used when we supply the input in the form of images. The last one is recurrent neural networks, and long short-term memory model is used when the input is in the form of time series. Also here in the column you can see the architecture of different artificial networks. So the first one, as you can see, is artificial neural network, where there is one column of input, second one is of hidden, and third one is output. Then you can see deep neural networks, and the last one would be the recurrent neural network. The hidden layer is recurrent in this particular method. The second application of deep learning is metagenomic gene prediction. Identification of genes directly from metagenomic fragments is an important task in annotating metagenomes. And metagenomic gene prediction is very important for identifying novel genes. However, due to the incomplete and fragmented nature of the metagenomic sequencing data, it is more complicated in metagenomes than in isolated genomes, and the assembly of metagenomes is often not available. Therefore, it is important to develop the computational methods for identifying the coding open reading frames from the metagenomics short reads. One way is to bypass the assembly and find the genes directly from these short reads. Some scientists propose a deep stack network learning model, which is named as metagenomic fragments with deep learning or meta MFDL, to predict metagenomic genes by fusing multiple characteristics of short reads, such as monoamine acid usage, open reading frame length coverage, and Z-curve features. Meanwhile, bacterial antimicrobial resistance is usually genetically encoded, and it is urgent to identify resistance genes in metagenomic samples. Therefore, the application of deep learning in identifying antibiotics is very important. The third application of deep learning is microbiome-wide association studies. These studies are to predict the relationship between the microbiome and the disease state. Microbiome-wide association studies have established that numerous diseases are associated with changes in the microbiota. Microbiome-wide association studies are similar to genome-wide association studies. They aim to identify microbial species, genes, or metabolites associated with the disease phenotype, which often involve feature selection and classification. Since they involve feature selection and classification, many feature selection methods have been proposed for the microarray gene expression and mass spectrometry based proteomics data to identify disease associated genes or the proteins. When algorithms are applied to these high dimensional data, a critical problem that is known as dimensionality comes up. Dimensionality reduction is one of the powerful ways to address the issue. In this regard, scientists have found that traditional machine learning methods were powerful classifiers when the data was limited. Also, the phylogenetic knowledge could improve the performance of the models. Therefore, few scientists have come up with two methods. One is known as autoencoder, and the second one is called deep forest. These both methods belong to the deep learning, and they are very successful for carrying out microbiome-wide association studies. Alright, let's get started with the challenges of deep learning applications in microbiome. First one is black box. It means that high accuracy deriving from the increased model complexity often comes at the price of loss of interpretability. It means that we are not sure after applying deep learning that if we are interpreting our results correctly or not. It is believed that there are two major reasons why the interpretable model is vital for bioinformatics and not only microbial data analysis. First one is understanding how predictions are made is essential to identify mistakes or biases in the input data when the model is trained. Second, deep learning could learn novel patterns from the massive biological data, which will guide meaningful insights if the model can be interpreted. So, therefore, interpretability is crucial in the field of plant and human health. Second challenge with the deep learning is large amount of training data. Deep neural network requires a lot of computing resources, just like this figure. If you want to dive down into the bottom of this iceberg, you need more oxygen as your resource. Similarly, if you want to dig more data, it could be computationally intensive and time consuming, as well as it also requires graphic processing units. A large number of data is required to train the neural networks. However, due to privacy concerns and high cost issues, it is impractical to obtain a large amount of biological data. Machine learning methods are superior in this regard. Therefore, when it is required, you could use machine learning methods. Otherwise, if it is necessary, you should opt for deep learning methods. The third problem is model selection and hyperparameter tuning. At present, there are 9 types of deep neural networks, and the practitioners and researchers propose a growing number of new models. However, each model is varying in different scenarios, and it is not easy and direct to choose a deep learning architecture for the specific task. There are many software libraries which are available now, such as TensorFlow, Keras, CNTK, and PyTorch. However, despite the availability of such libraries and tools, the task of picking the right neural network model and its hyperparameters are usually complex. As you can see in this figure, that there are 3 hyperparameters, uh, number of given by the number of layers and number of neurons, and on the right side you can see the scores. So it seems to you that 92% maybe is the best one. However, you cannot tell by just looking at the numbers because it's a subjective choice. So it depends on your final goal, what you are looking for. Therefore, for one person could be 92% model would be the best. However, for the second might be preferring 85% model. So it's complex process. Now that we have finished the topic, I hope that we have been able to understand basics of deep learning in microbial data analysis, why it is important, how we can use it, and the challenges in the future. Thank you.